Hello everyone, and thank you again for joining us, and thank you, Wilson, for the introduction. This year has been really a transformational year in cancer genomics with the introduction of the Revio system, which you see on the right, which really allows for the first time the true complete interrogations of genomes, epigenomes, and transcriptomes. And we know that cancer biology is incredibly complex, and building on the success of the completion of the human genome, we've now taken this technology and now have the capability with regards to the throughput and depth of sequencing to really interrogate cancer genomics and cancer biology. And the advantages of our long-read sequencing is really can be summarized in two main pieces, two main points. One, as you see on the left, that we have long reads. Those are 100 times longer than next generation sequencing, the short read sequencing from Illumina, and really allows us to capture large structural events, things like structural variation, fusions, and isoforms that short reads simply cannot. On the right, you also see a graph that shows the accuracy of our long read sequencing, which is very important. It is equivalent or exceeding that of current short read technologies and exceeds that of other long reads technologies by quite a bit. So really allows us to really look at large structural variation, but also the small mutations such as SNVs that are really important in cancer. So again, the length of the reads and the accuracy are incredibly important when you look at the genome or the transcriptome. So cancer is a disease of the genome. It's driven by mutations, but most of the dysregulation, as we know, really resides and can be detected at the level of RNA. So we can look at things like gene expression changes in tumors, but also RNA isoforms, fusions that are very common in cancers, in some cancers, and things like express mutations, where we look at, which will be very important actually for today's talk, where we're looking at the mutations actually expressed in driving tumor initiation, progression. So continuing, when we perform RNA sequencing, we can compare the way that we analyze with short reads versus long reads. And with short reads, of course, we have short fragments. An average transcript is about 2 to 2.5 kb, and the short sequencing fragments from short read sequencing are about 200 base pairs. So what we're able to do is really capture small fragments and reconstruct, assemble the transcriptome from that. And with that, we really can assemble only 20 to 40% of the transcriptome. So we lose a lot of information. On the right, you see with long read sequencing, we're able to really sequence full molecules. So we're able to get the 5' to 3' and really look at biology or the molecule as it is in completion. So we don't lose any information and we get a complete view of the cancer transcriptome. And again, apologies for the technical difficulties here. So one great example is alternative splicing. We know that genes do not express one gene, one transcript. They express a few or sometimes many. So one gene results in several RNA isoforms that result in different proteins with different functions. And it's really only with long read sequencing that we're able to truly capture the diversity of those transcripts in cancer and any tissue. One key publication that came out last year in Science Advances did this comparison for the first time, to really looking at tumor samples and sequencing with short reads and long reads. And as you can see on the right, found using long reads, tens of thousands of novel isoforms that were completely absent or missed by short read sequencing. So again, one example of what the extra information that we get when we use the correct technology. Recently, we've partnered with the Broad Institute to really commercialize and kit a what we call mass Iso-Seq, which is a protocol to take this to the single cell level. So any of you that have used 10x Genomics platform, you can now perform long read single cell sequencing using PacBio with our accurate long reads downstream of the 10x platform. You can see on the left that with regards to gene expression, we can, because of our throughput of this method, we can cluster cells in tumors or any other tissues just as well as short reads can. But also on the right, as I said, we can capture this information that is missed by short reads and layer this information such as isoforms, fusions, and mutations onto these t-SNE plots, these single cell plots, and really understand the true heterogeneity, the both cellular and molecular heterogeneity of tissues, and that is especially important in cancer. Another preprint that came out from Switzerland just earlier this year really showed, did a similar comparison at the single cell level using this methodology that I just showed, and really showed that beyond just gene expression, using long-read, accurate long-read sequencing, you can do really 4 things. And I encourage all of you to take down the reference on the bottom and read this paper. We don't have time to go through the whole publications, but I just want to highlight 4 things that they looked at and really found. One is that, again, you can get equivalent cell type classification as you do with short-read single-cell sequencing. 2, you can discover novel isoforms, as we just saw from the previous publication. You can also detect known and novel fusions at the single-cell level that are very important in cancer. And again, because of the accuracy of our sequencing, you can really look at expressed mutations. So look at the mutations at the level of RNA with very, very high accuracy. So you really are covering all of the different levels of RNA dysregulation that happen in cancer. And those different levels of regulation are incredibly important when we look at neoantigens in immuno-oncology, so which will be the topic of today's talk. So neoantigens are what is presented by tumor cells to the immune system and act as targets for immunotherapies and cancer vaccines. And until now, because of the limitations of short-read sequencing, the neoantigen discovery has really been limited to SNVs, so really point mutations. Now we can really look at all classes, all variants, all types of neoantigens with long-read sequencing, such as isoforms, fusions, as well as SNVs. And these isoforms and fusions especially have really been known to be potent neoantigens in cancer and immuno-oncology. So we really believe that this platform, this technology can really be the way to perform neoantigen discovery in the future. So with that, thank you very much. Really looking forward to the talk following me. And again, apologize for the technical difficulty that we just encountered. Dr. Li, the stage is yours.
Okay, thank you. Thank you very much for providing me with the opportunity to present our recent work. My name is Zhongxiao Li. I am a PhD candidate at King Abdullah University of Science and Technology, or KAUST, in Saudi Arabia. Today, I'm going to present our recent work powered by PacBio Iso-Seq, and the project is entitled The Full-Length Transcriptomic Atlas of Human Colorectal Cancer from Single-Cell Isoform Sequencing. This work is a collaboration between the Computational Bioscience Research Center here at KAUST and the National University of Singapore, the Cancer Science Institute of Singapore. So first, let me give a brief introduction to the background of this research project. As we all know, gene expression goes from DNA to RNA and then to the protein. At the RNA level, the transcriptional and post-transcriptional regulation of gene expression include alternative TSS, alternative splicing, and alternative polyadenylation. These types of regulations have been essential in controlling the structure, the sequence, as well as the quantity of the mRNA product, and have profound implications in normal cellular functions, as well as in diseases such as cancer. Long-read sequencing, because it can sequence isoforms in full length, is the most accurate way of detecting these kinds of regulations, and it is one of the reasons that it was designated as the method of the year in 2022 by Nature Methods. Recently, our group systematically studied the splice events in the 3' UTR regions of RNA transcripts of the oncogenes using TCGA and GTAG's short-read RNA-seq data, and have shown important implications of such events in tumorigenesis. For example, a splice event in the 3' UTR region of an important oncogene in the Wnt signaling pathway, CTNNB1, which is upregulated in colorectal and liver cancer, may promote its mRNA's export from the nucleus to the cytosol, and therefore subsequently contributes to its overexpression. This motivates us to build a full-length transcriptomic atlas of human cancer from single-cell sequencing in order to obtain a resource that is more beneficial to such studies. We obtained tumor and adjacent normal samples from 12 colorectal cancer patients from the National University Hospital of Singapore. Their RNA was extracted and reverse transcribed into cDNA and then tagged with cell barcode by the 10x protocol. The portion without fragmentation then underwent PacBio long-read sequencing, while the portion with fragmentation then underwent Illumina short-read sequencing. While the Illumina sequencing part was mainly responsible for cell typing and gene-level expression quantification, the PacBio part was responsible for full-length transcripts characterization and isoform-level expression quantification. The common cell barcodes of the two parts are then used to connect the two components of the atlas. This figure gives an overview of the data analysis pipeline of this project. First, I will introduce the result of the pre-processing steps of the two sequencing methods, which include cell type identification and full-length transcript isoform calling, and the connection between the two sequencing methods via common cell barcodes. To build the short-read part of the atlas, we used Cell Ranger to quantify the gene-level expression of the samples that underwent Illumina short-read sequencing. We independently processed the normal samples and the tumor samples, and then they were integrated with a colorectal cancer single-cell atlas from the Broad Institute containing 300,000 cells. We classified the cell types of our in-house data by transferring the cell labels from the Broad's data to our in-house data. This t-SNE map visualizes the integrated dataset. The Illumina component of our atlas has integrated very well with Broad's, and in this way, we can identify a total of 20 cell types, including the epithelial cells, the immuno and stromal cells of the tumor microenvironment. Next, we move on to the long-read part of the atlas. Using Candice3, we classified the identified isoforms in our PacBio dataset into 4 main categories by comparing them against the reference transcriptome. These include FSM and ISM for transcripts with only known splice junctions, and NIC, which contains novel combinations of known splice sites, as well as NNC, which contains novel splice sites. We observed a higher percentage of NIC and NNC isoforms in the tumor epithelial cells as compared to other cell types, which indicates transcriptomic dysregulation among the tumor epithelial cells. Overall, more than 60% of the genes were detected for more than one isoform, and almost all FSM and ISM isoforms were having only the junctions known in the TCGA colorectal cancer samples, while NIC and NNC had a much higher proportion of novel junctions. This last figure shows the isoforms detected for the gene epicam. Although being an important epithelial cell marker, we still detected 12 NIC and 25 NNC isoforms, with 28 novel splice sites in total. Next, we evaluated the correlation between the Illumina component and the PacBio component of our atlas. In total, there are about 2,000 overlapping cell barcodes between the short-read and long-read sequencing. For all of the cell barcodes detected in PacBio, only the cells in this intersection can we determine the cell type from which the isoform comes from. Most cells are the epithelial cells, including both the tumor and normal epithelial cells, followed by several immuno and stromal cell types. Almost all genes covered by the long-read sequencing have also been covered by the short-read sequencing. Common genes were most abundant in normal and tumor epithelial cells. Correlation of the gene-level expression is high among the epithelial cell types, followed by plasma and CD4+ T cells. To fully utilize our full-length single-cell colorectal cancer atlas, we next performed analyses that are so far only possible with long-read or a matched short-read and long-read sequencing experiment. This includes simultaneous inspection of differential gene expression and differential isoform expression. This also includes analysis of isoforms associated with particular epithelial cell lineages. This also includes the analysis of splicing events associated with each isoform, especially when there are more than one alternative splicing events associated with the isoform and how the coupling of them happens. We also analyzed the RNA editing associated with different isoforms. Finally, because we have the sequences of the identified isoforms, we predicted their open reading frames and performed proteomics analysis of those coding isoforms. First, let's start with differential gene and isoform expression. In contrast to single-cell short-read sequencing, as it can only tell differential gene expression, or DGE, we particularly investigated differential isoform usage across the different cell types. In particular, we investigated the dysregulated transcript structures, or DTS, across tumor and normal epithelial cells, which is defined as genes having different allocations of their isoforms. We observed that genes with DTS are more likely to be also a DGE, suggesting that they are both under regulation at the isoform level and the gene level. The genes with DTS are more enriched in pathways like mRNA binding and RNA catabolic process, which suggests that there is transcriptomic autoregulation. There were also several immunological pathways that were also enriched, such as immunoglobulin binding, which suggests that there is dysregulated immunoactivity in those tumor cells. We further analyzed the alternative splicing events associated with the isoforms, and we in total analyzed 7 types of such events, and all of them are listed here. We recorded the isoforms that were implicated in a particular event among the diagonal entries of this heat map. And for the isoforms that are associated with more than one such event, we record them in the off-diagonal entries of this heat map. In particular, we found out that the DTS genes were particularly enriched in alternative splicing categories when there is coupling between, for example, alternative first exon and retained intron, or when there is coupling between alternative first exon and alternative 3' splice sites, or alternative 3' splice sites and retained intron. We point out that studying this kind of coupling between alternative splicing events is only possible with long-read sequencing, which can sequence the full-length isoforms. We show through 2 examples where there is coupling between alternative splicing events. For example, the gene ARGHDIA is only possible to have a downstream retained intron event once it is utilizing a distal first exon. As another example, the gene TMEM259 is only possible to retain an upstream intron once it is utilizing a proximal alternative 3' splice site. Next, we analyze the dysregulated transcript structures among the epithelial lineages. The intestinal epithelium is the fastest self-renewing tissue in mammals, which contains epithelial cells with diverse differentiation statuses as well as proliferation activities. As epithelial cells are the most abundant cell type identified in our atlas, we decided to delve into this cell type by further categorizing the cells into subclusters that correspond to different lineages of those epithelial cells. These include stem or transit amplifying cells located in the crypt, and the more differentiated enterocytes and goblet cells located in the villus. In total, we categorized 3 stem cell subpopulations, one goblet cell lineage, one anthrocyte lineage, as well as another lineage with positive expression of bestrophin-4, which is a recently discovered epithelial subtype due to single-cell sequencing. We investigated the isoforms that were differentially expressed in those lineages. As an example, the gene KRT8 has 3 typical isoforms with protein products of different sizes. Earlier studies had observed a preferential expression of the largest isoform in SMAD4 positive colon cell lines as compared to SMAD4 negative colon cell lines. In our dataset, we also observed an overexpression of this isoform among the tumor stem cell types as compared to the normal differentiated cell types. And there is a negative correlation of this isoform's usage to SMAD4 expression among those cell types. Post-transcriptional RNA modifications on single nucleotides could also impact the mRNA sequence and the expression without altering the transcript structures themselves. The most notable example is the ADAR-mediated A to I RNA editing. Because A base pairs with U, but I base pairs with C, this can potentially alter the mRNA coding sequences. We investigated the RNA editing events detected in our long-read atlas. In particular, we computed an RNA editing level per site score, or REPS, by computing the ratio of the edited reads to all reads at a particular site, regardless of the isoform that it comes from. We also computed an RNA editing level per isoform score, or REPI, by computing the ratio of the edited reads to all of the reads at a particular site, but for each isoform individually. Through the REPS analysis, we found a previously recognized editing site of CDK13, which is upregulated in the tumor epithelial cells as compared to the normal epithelial cells. We also observed upregulation of RNA editing events in other genes, such as CYP2R1 and ANKRD40CL, as well as NEAT1. There are some other events that were downregulated in tumor compared to the normal epithelial cells. The examples include the genes such as ANDOLF. Due to the sparsity of the long-read sequencing data, we only managed to observe a few multi-isoform genes with more than one of their isoforms edited. Therefore, we only computed the REPI score for these particular 3 genes, which are CDK13, ANKRD40CL, as well as NEAT1, because they have the most number of edited isoforms. Notably, the 4 isoforms of NEAT1 and ANKR, as well as the 5 isoforms of CDK13, all displayed heterogeneous isoform-resolved RNA editing levels, which suggests that the RNA editing levels might be different for each isoform. Finally, we predicted the open reading frames of our detected isoforms. Overall, the majority of the isoforms were coding transcripts, with NIC and NNC having a slightly lower coding potential than the other types. The open reading frames typically starts with a canonical start codon, while the ISM category usually terminates prematurely, which indicates their incomplete identification of the isoform. The novel categories are more likely to be targets of nonsense-mediated decay. Comparing with the UniProt sequences, the novel categories are also more likely to produce novel peptides that were not seen in the UniProt database. We searched against orthogonal mass spectrometry data of those predicted open reading frame sequences. 26 and 44 novel proteins from the NIC and NNC category were directly supported by orthogonal mass spectrometry data in CPTAC or our in-house data. There were also more MS-supported novel isoforms from tumor epithelial cells and tumor samples as compared to their normal counterparts, suggesting that transcriptomic dysregulation in tumor cells could result in increased abnormalities of the proteome. Since we already obtained the predicted open reading frame sequences of the isoforms, in this last section, I'm going to introduce our proof of principle workflow of utilizing PacBio long-read sequencing to derive recurrent tumor-specific neoantigens for colorectal cancer and show how they can be useful for cancer vaccine development. Neoantigens or neoepitopes in tumor can come from a variety of different sources. These include somatic mutations, alternative transcript start sites, alternative splicing and RNA editing, as well as gene fusion. This also includes alternative translation, which may directly alter the translated amino acid sequences themselves. Such neoantigens can be exploited to boost the patient's immune system using cancer vaccines, such that the cytotoxic T cells of those patients will recognize the tumor cells whose neoantigens could be presented by their MHC1 molecules. Recently, huge development has taken place in the field of cancer vaccines. For example, Moderna's mRNA-4157 cancer vaccine has been shown to be very effective in melanoma in combination with PD-1 immunotherapy. Earlier this year, researchers from Sloan have demonstrated in their phase 1 clinical trial the effectiveness of a neoantigen-based cancer vaccine in pancreatic cancer. However, it should be noted that the neoantigen sources from both of the two clinical trials are from somatic mutations. Although some somatic mutations are conserved across patients, a lot of those passenger mutations may be specific to particular patients. In this way, both studies required personalized cancer vaccine design. However, alternative splicing-derived neoantigens have been largely underexplored in previous studies, except that in 2021, Wang and their colleagues, using short-read data, investigated a particular type of alternative splicing event called excitrons for their potential in the production of neoantigens in cancer. Excitrons are cryptic introns that falls completely within the protein coding exon. Because of this structural simplicity, it is possible to be studied using short-read sequencing. There are several advantages of the neoepitopes that are derived from aberrant splicing. On the one hand, alternative splicing, especially when it alters the coding sequence of a portion of the transcript, may produce a lot more neoantigens per event as compared to somatic mutations. On the other hand, such events may also be more likely to be shared across patients, as most splicing occurs at canonical splice sites, the JOAJ motif. Because of the existence of such aberrant splicing-derived neoantigens, we speculate that finding such neoantigens can be a potentially very important clinical application of single-cell long-read sequencing. This is because using long-read, which can cover a much greater portion of the transcript, can detect more alternative splicing events instead of just the excitron-like events in the previous studies. Plus, when the isoforms are sequenced in full length, we can more easily predict their open reading frames. The single-cell information preserved during sequencing is also very crucial in that we hope the selected neoepitopes to come from the solid tumor rather than other cells in the tumor microenvironment. We therefore designed the following proof of principle workflow to select a panel of high-confident neoantigens from recurrent tumor-specific novel isoforms in colorectal cancer patients. We first selected the NIC and NNC isoforms exclusively expressed in the tumor epithelial cells from the long-read component of our atlas. We use string type to quantify their expression levels in the TCGA colorectal cancer patient samples. We required the isoforms expression levels in the normal samples to be less than 1 ppm to ensure tumor specificity. We additionally required those isoforms observation in orthogonal mass spectrometry data to further increase their likelihood of existence. Using the predicted open reading frame sequences of those isoforms, we selected 9 mers that were not observed in the UniProt protein sequences, as well as in the non-isoforms open reading frames. We then typed the TCGA colorectal cancer patients' HLA alleles using their short-read sequencing data and used NetMHC-PAN to evaluate their binding affinities to those neoantigens. Because the number of neoantigens that can be filled into a cancer vaccine's payload is limited, we need to curate a subset of those neoantigens that work as diverse a group of patients as possible. Therefore, we applied a greedy algorithm to optimize a panel of 24 neoantigens that have a high coverage of the target patient's group's HLA alleles. So this figure illustrates the selection process. So these are the tumor-specific recurrent isoforms, and they can produce neoantigens, which are listed here. And each of their neoantigen is evaluated by NetMHC-PAN, their binding affinity to the patient's HLA alleles. And if a line is drawn between the neoantigen and a specific HLA allele, it means that they have strong binding affinity. So on the first iteration, a specific neoantigen is selected, for example, this one, because it has a very high coverage of those HLA alleles. And then on the next iteration, a next neoepitope is selected because it has the most number of binding, strong binding HLA alleles for the rest of the HLA alleles, and so on and so forth until all of the patients are covered with a significant number of neoepitopes. So this left panel displays our optimized 24 neoantigen panel with their binding affinity to the TCGA patient's alleles. Each neoepitope will preferentially bind to a specific type of HLA allele. But once aggregated, this last row shows that most HLA alleles have high binding affinity to at least one of them. In total, there are 65% of patients who have at least one neoepitope in the panel from an isoform that confidently expressed in their TCGA sample that also strongly binds to one of their HLA alleles. On the right-hand side, in this dot plot, we show the distribution of the highest binding affinity of each HLA allele or each patient to the 24 neoepitope panel. Each dot represents an HLA allele or a patient, and the vertical axis shows their highest binding affinity to a particular neoepitope in the panel. All HLA alleles have at least weak binding affinity to the neoepitopes, and all patients, because it is aggregated across their HLA alleles, all patients have at least one neoepitope with strong binding affinity. The median number of the neoepitopes that bind strongly to an allele is 3 for HLA type A, and 4 for HLA type B, and 5 for HLA type C. Once aggregated for each patient, the median number is above 10, which means that 50% of the patients have at least 10 strong binding neoepitopes to one of their HLA alleles. We further validated some of the novel isoforms where the neoepitopes in the panel come from by first validating the unique splice junction sequences through Sanger sequencing. And secondly, we used RT-PCR to validate the existence of their final mRNA product. In this way, we further improved the credibility of those recurrent isoforms' existence. Finally, these two examples illustrate the origin of the neoepitopes in the panel. In the first example, the gene STMN3 utilizes an alternative first exon, and the neoantigen comes from the novel ORF caused by the novel junction. In the second example, the gene CPNE7 also utilizes an alternative first exon, However, this time the neoepitope comes from the novel open reading frame introduced in the alternative first exon. So to conclude, in this project, we have built a full-length transcriptomic atlas of human colorectal cancer by integrating the short-read and PacBio long-read single-cell RNA-seq data. The atlas has unveiled genes and transcripts associated with tumor epithelial cells, as well as different epithelial cell lineages and subpopulations. By integrating the novel open reading frames from recurrent tumor-specific transcripts and mass spectrometry data, we were able to design a panel of 24 neoepitopes, which have strong HLA binding affinities that can be exploited for the development of neoantigen-based cancer vaccines. Finally, I would like to thank my own group at the Computational Bioscience Research Center at KAUST, and this is where the major analysis of this paper took place. I would also like to thank the Bioscience Core Lab of KAUST, which provides us with the PacBio SQL2 platform. I would also like to acknowledge our collaborators at the Cancer Science Institute of Singapore and the clinicians from the National University Health System of Singapore for their collection and preparation of the clinical samples, as well as the construction of the 10x single-cell library. Thank you very much for your time and attention, and I'm now very happy to take any questions from the audience. Thank you very much. So here is the link to our preprint, and I have given this QR code here. So in case you would like to save for your later reference.